Hello, everybody. I wanted to welcome you all to this video. Thank you for choosing to press play on it. My name is Michael Luella, and I am using they, them, and their pronouns. And I am speaking to you from Seattle, Washington, in the US of A. And I work for defeatHIV, which is one of the 6 Martin Delaney collaboratories that is working towards an HIV cure. And I'm their community engagement project manager. And with me are...
I'm Lynda Dee from the DARE CAB. I am the co-chair of the DARE CAB, and I live in Baltimore, Maryland, and I'm from AIDS Action Baltimore, which is my home organization.
And I'm Deeks. I'm a clinical investigator in San Francisco, and I help lead with Lynda and others the DARE collaboratory. And our goal is to develop an immunotherapy for an HIV cure.
Awesome. Thank you so much. And today our topic is really about the future that we see ahead for cure research for HIV. I call it the roads ahead for this. And really what I want to emphasize is what we think people need to know. You know, what's the important stuff? There's lots of things that we could talk about, but what do you think it's important for people to know? And that's where I wanted to start off. And so, Lynda, I know you wanted to start off talking some things about the community. So what do you think it's important for the community to know about the community?
Well, first, Michael, thank you so much for inviting me, and I'm really glad that Steve was able to join us.
Me too.
But, you know, for me, I've been doing this work for about 33 years, and I came through learning about drugs and research and trial design and all of that interesting stuff many years ago when we were looking at antiviral research that actually, I mean, look how far we've come. It's unbelievable what the strides we've made. That essentially people are taking one pill once a day to survive and live pretty normal lives. So we made a lot of mistakes in those days as far as who we engaged in the trials, who we did outreach to, who was included and who wasn't, and that includes the United States and in the South, you know, in developing countries and even, you know, mid-level countries. I mean, I think that we could do better this time, and I think one of my goals and the goal of our CAB is to make sure we do better this time, make sure more people are included, and, you know, we need to work on the science and on community issues as well.
So the populations that you think that we missed in the initial rollout of HIV research, what would those populations be specifically that we should focus on for cure?
Well, I think not only should we focus, but we have to make sure that we engage them and educate them and let them know what this stuff is about and, you know, what's available and what's not. So we need to let them know, just like we've done in the PrEP arena, let people know that it's available, that it works, and, you know, how you get your hands on it. And in cure, it's much more difficult than that. It's really a complicated area of science. And so we have to be sure that women know about it, that people of color know about it, both Latinx people, African American people, and again, that people in the developing world, in the South, know about this and have access and don't have to wait 20 years till they're included in this deal.
Lynda, did you know, just to bring this up, Gilead funded a community piece that was focused on HIV cure in the South. It was a qualitative piece of research. I've not seen the end results of that, but I remember when they first started off, we invited them up to Seattle to present at our Cell and Gene Therapy conference. And it seemed like it was really interesting because it was going to give us a sort of initial impression of the needs of the South in terms of the populations living with HIV and their attitudes and opinions about HIV cure. I haven't heard anything from that, but I think it sounds like it would be a good step right towards what you were saying in terms of what we need to focus on, because it would be giving us some concrete data to work from.
Yeah, you know, I think that Steve has been involved in some endeavors in that regard, that Gates is reaching out to do that. I mean, I think that oftentimes, you know, it's easier to get this kind of stuff done in the South than it is in the United States. It's very interesting. I was thinking about this the other day, you know, contemplating the 2020 AIDS back in the United States. And I thought about the first conference in South Africa and what that was like and how that absolutely changed the world as far as access to everything for people in the South. And I mean, you know, researchers from the North started working there. I mean, it's been the joy of many of their lives, you know, to do the work in this, you know, the sub-Saharan Africa, for instance. Anyway, you know, I think that we don't have to rattle their cages about that as much as we do oftentimes in the United States. I mean, I think people are interested in doing that. I think the problem with cure for the South is going to be that it'll be scalable, that it'll be simple enough that it can actually happen in those countries. They may even have better access than the United States, where everything costs $1 million. So.
Steve, what do you think about that?
Well, you guys are covering a fair amount of stuff. You know, getting back to this issue, and Linda's been harping on me this for many years. You know, about the populations who were engaging in these early clinical trials. They need to reflect the populations that are ultimately going to require to be cured. I mean, the fundamental issue is that I think that the greatest unmet need in terms of H therapeutics is those 50% of the world's population who are not accessing therapy for whatever reason. And most are not accessing therapy because they're basically disenfranchised from healthcare. They lack access. Some of these drugs are showing excess amount of toxicities in specific populations. For example, the integrase inhibitors and weight gain in young African women. So, you know, my 99% of the people who I've enrolled in my clinical trials over the past 25 years look a lot like you, Michael.
Yeah.
And
they're bald?
And the bald, yes, definitely bald.
They are now.
Other characteristics. But, you know, and so I was, you know, I initially thought people, drugs are drugs, until we did a study with Eileen Scully a few years ago, in which we very carefully studied the impact of a curative intervention on men and women, and found that there are actually quite marked differences between men and women in terms of the size of the reservoir, where it's at, how active it is. So I'm a convert. I now appreciate that clinical studies need done in people who are going to require theirs in the long run. How to operationalize that's a whole different issue. But I'm on board, and we need to do better. And I think we're doing better in this regard.
Right. Yeah, it's been difficult for me when I've been trying to engage people around gene therapy and HIV cure, just because there's two levels to that. There's the HIV cure part, and then there's just the gene therapy part. And they come packed with so many triggers that I was not expecting, so that when I was engaging different populations, I realized I had to take different tacks and try to find different ways to bring in ideas and find different metaphors to talk about things. And when you found a metaphor, you could tell, because that's when people would sort of light up. But ultimately, I felt like we needed to do a lot more for African Americans in the US, but then I also was worried about Africans in the continent of Africa all over. I thought, how do we describe bone marrow stem cell transplants in different languages in Africa? How do we start to talk about what it means to edit a cell and add some DNA or to subtract some DNA? And how do we do that in a simple way that we can get them on board for a potential cure? And I'm just happy to know that the NIH and the Bill and Melinda Gates Foundation has decided to put money towards that, which is going to include engagement, because those limited resource settings that they are focusing on are going to need to be the ones to do that for us. I can't create those materials here. I could love to help, but I can't do it myself. They're going to have to know their people, and they're going to have to know what works for their people. And that's going to be different all around the continent and then all around the world. I also find that treatment access comes into play here, because we still have people not on treatment. And so it's like a luxury to be able to talk about cure when you're on a pill a day or when you have your medical care. But if you still don't have ARVs accessible, HIV is still a death sentence. And are you going to be wanting a cure? You're going to want it maybe in that abstract sense, but really, how can you be even asking for money for cure when we need to get those people that access to that life-saving medicine still? And I find that depending on who I'm talking to, I need to frame and reframe cure as a part of care. I have to change the U of cure into an A and make it as a continuum, so cure becomes a part of care, so that when we do, if we do ever find a strategy that works and have a technology that works, that it seems a natural consequence of getting involved in care. I don't know, maybe it's idealistic.
And, that's really very important, because in the United States, we have a tendency to silo things. We put prevention here, care here. It's ridiculous. And, once we got PEPFAR going, I mean, they don't do that. They integrate things, and that's what we should do. But it's interesting, the way you describe that reminds me of Steve's scope cohort. I mean, he treated those people for years. They've been in trials with him. They're absolute partners. I mean, they've worked together for years, and now, they're entering the cure phase of this. So it's actually exactly the continuum that you described.
Right. And I think it's important for the people who are not necessarily intimately involved with research begin to realize that and figure out ways that they might be able to stay informed and add it in. So if you're a case manager and you're hearing this, like, how can you begin to add cure into the paradigm of what you may already know about treatment and the options that are coming there? And that's not because we want to get your caseload to become research subjects. It's really more about helping to pave the way, because it's going to take everybody to help pave the way to a cure, just like it took everybody to start getting PrEP rolled out, and we haven't even really finished that. But that's one of my other concerns, I think. We have lessons from the past. Do you think that we're learning those and that we're going to be able to change the way we get to roll out cure and the way we get to approach research a cure? We know things about populations. We know things about access. Are we prepared for cure? Like, I guess let me say it this way. What I'm afraid of is that we're going to get a cure and it's going to be too expensive for the people who need it the most. So it's going to be like Hep C medicines, where we're going to have this thing come out and we're going to see what's going to look like a greedy pharmaceutical company asking for a lot more than maybe the research and design actually calls for. And yet nothing's going to change, and that's just the way it's going to be. And then we'll scramble around, and there'll be 5 years before we get something actualized to finally get it to those populations. And it still will be, in the US at least, a white guys get it first, because they're the ones that are most connected and they've been involved. And I want to break that in some way. I just don't know what to do to make that happen.
What you need to think about is not just the end. You need to think about the beginning of that too. It's not that, for instance, in the United States, that people of color are not interested in this. They just don't know about it, because they're always the last ones to get the information, for a myriad of reasons. So if we get them in the front of the line with everybody else to start with, when we get to the back of the line, maybe they'll have some advocates too. I mean, they certainly do now. But, this will be just as important as the other services, and they'll be in line with everybody else. As far as a pharmaceutical company, a lot of companies go, I mean, I've been working on that. Martin Delaney drugged me into that 100 years ago, and it is the most frustrating work that I do, especially in the United States, where it's against the law to, you can get in lots of trouble for not making all the money in the world for your stakeholders, but nothing happens to you if your drug is too expensive and people die as a result. I mean, it's like, it's don't get me started. But I think that we have to be cognizant of that up front, and people like me will continue to torture people like Carl Diefenbach to make sure that he's making, the monoclonal antibodies, for instance. He's working out things with different companies that'll make them accessible to people in the United States. Forget about the rest of the world, but the rest of the world certainly. But I mean, it's sometimes worse here than in other countries where they don't have the patent restrictions that we do.
One of the things.
Let
Go
me
ahead.
comment, though, Michael, about some of these issues, because I don't think that they're necessarily as in conflict as they used to be. All
Okay.
right. So let me just, in Seattle, your team is working with some tech stuff, some seriously high-tech, non-scalable,
Yep.
$1 million interventions, right?
Yeah.
You guys are talking about taking stem cells and expanding them and knocking out genes and knocking in genes and giving people chemotherapy and then pouring these things in the vein, stopping therapy. I mean, stuff that happens now in cancer therapy where it works, but it costs $1 million a treatment.
Right.
Okay?
Right.
Somewhat, and it's primarily being done in older white men, I am sure.
Mm-hmm.
Okay? We're doing the same. These things can only be done in these really urban centers that are really well resourced, have lots of experience, typically with highly engaged patient populations that have been involved in research for quite some time. And that's kind of at the same time, we got the Gates Foundation and the IAS and the Sunnylands Initiative, right, that we'll talk about at this meeting in San Francisco. We're out there on the front lines, working with our colleagues and partners in Africa and asking questions, what do people need? And basically what I think people need, but what I've been told, I mean, what the ideal thing is, so people around the world, they can access treatment and they take the pills, they do well. People who can't access treatment, that's a challenging population. Ultimately, what's going to really change the world is if you find people with HIV, you give them a shot in the arm, and it works, and you go away, and they're cured. But there is a direct line, in my mind, from the work you're doing in Seattle to that one-shot cure that will actually impact globally. We won't get to that one-shot cure unless we do this high-end stuff first.
Mm-hmm.
So they're well aligned. So I think we can do the really deeply intensive study, prove that we can get people, begin to operationalize it, optimize it, and eventually figure a way to make it scalable. And do that while we're having discussions globally with impacted people so that we know what will be desired, because if it's desired, then it will be used. If it's used, it will be impactful. These things are happening simultaneously.
Mm-hmm. I agree with you.
So I'm more optimistic that we've learned from our mistakes.
Good. Well, I'm glad you are.
Well, that's what I said about, sometimes it's easier to be involved in the South than it is with different populations in the United States, because people know that it's possible to do the work there and that the people are engaged, and it's really very heartening to see.
And so we've started talking a little bit about the science, and you brought up the gene therapy. Where do we see the science going for the next, let's say 5 years? What do you see happening? And you can focus on DARE if you want, since you're probably more intimately involved with that. But anywhere, like I'm interested to know. Like, it seems like everything is in still phase 1, small pilot studies or lab basic science, and it's hard to get an idea of where I should put my eggs, so to speak. Which basket should I put my eggs in? I want an egg in every basket, actually. But it seems like sometimes we get a lot of gene therapy because it's sexy in the news, but I don't hear as much about block and lock. But maybe that is actually going to be a way that is valuable for us. And so all of them are going to be valuable because they all are going to help us understand HIV and the immune system. But what ones might we want to be paying attention to? Or let me say, what in each area should we be paying attention to? We should be paying attention to all of them, but what should we be keeping our eyes peeled for? And these are opinions, not facts.
Multiple shots on goal. That's where we are right now.
Yeah.
It's like with the COVID vaccines, right? There's 6 or 7 of these things moving to phase 3. No one knows what. Multiple shots on goal. So there are 5 trajectories that each have their pros, each have their cons, that are actually not as siloed as they used to be because we're combining them now. And I don't know, Micah, you want to go through one by one and talk about the state of the art? I mean, we can do that.
I think that would be at least illuminating, not only for us, just to make sure we're talking about the same things, but for the people who may not know, and they may not ever get to go to a pre-conference because they're not paying to go to AIDS 2020. So this will be a free way for them to get that for us. So let's go through.
All right, let me, so Lynda, maybe I'll talk about the science. You can comment a bit on the real world issues for each of the 5 trajectories, okay? So there's shock and kill. So I'm going to start with shock and kill because that was the first one that was started about a decade ago. This is the concept that the virus is hidden in these cells in the body, and we want to come in with a little bit of a nuclear bomb, explode the virus out of those cells so that the cells die and the virus dies. We have played around with this in the clinical trials for about several years. We've done 2 or 3 ourselves. We've gotten nowhere. Both of the studies have failed. We've shown that you can actually shock the virus. You can't kill the virus. But in the last year or so, 2 things have emerged that are very promising. One is some work that's being done by Tim Shacker, interleukin 15. He has some pilot data that interleukin 15, by turning on the immune system, can cause both the virus to come out and cells to die. And that's the first time that's been shown in humans.
Wow.
And 2, the team at the Emory and at the University of North Carolina have identified the SMAC mimetics, the shock drugs that are actually remarkably powerful in mice and monkeys. And so we got 2 agents now, interleukin 15 and the SMAC mimetic, which appear to suggest that at least the shock part might actually begin to work. We still have to figure out the kill part. But anyway, I think that's shock and kill is still very vital, and some progress being made.
That's good to know because the River study seemed to be the first big trial, and that didn't work. And there's all the other labs. It's good to hear someone say, "Well, it's still in the ball game."
I can tell you there's 20 or 30 failures, including the River study.
Mm-hmm.
These drugs have generally shown that you can actually cause modest reversal in people, but nothing like that's been seen with interleukin 15 or SMAC mimetics.
Awesome.
I hate this approach, okay? I
Okay.
think it's counterintuitive. So you've got this latent virus, it's not alive, or it's not kicking, and let's wake it up, okay? And then drop a nuclear bomb on it. Well, hello, I hate to be that patient, I mean, I like the approach of the immune system working through this, and somehow at the same time you're waking this up, having something that from the immune system that's going to counter this. But so far, and it could very well be that the drugs we've had for this have been really, really terrible. I mean, a lot of them have been old cancer drugs, extremely dangerous, with the possibility of giving people cancer down the road. I mean, well, what's the risk benefit of that? You could take 1 pill once a day, or maybe it doesn't make any sense to me to ask patients to go through that risk in the trials, and then have to drop some bomb when I like it. I'll give you a segue into the block and lock. Let's have our own body or interventions be able to keep the virus latent and make people, elite controllers so that their own bodies can do this without having to blow up. Use some nuclear option to try and do this and hope for the best.
So Lynda brings up block and lock, which is the opposite of shock and kill. And block and lock is, as Lynda said, you basically shut the virus down so deeply. It's I always think of it as like pouring concrete on the virus. It's basically locked in, it can't come out, and therefore, who cares? And we think this is plausible because if you look at people on long-term treatment, 20 years, there's evidence that it looks like the virus that persists that long is really in deep, deep latency. It doesn't come out. So there are host mechanisms that can do this. There are these group of so-called extraordinary or exceptional controllers, these rare people that are being talked about a lot now.
Right.
If you look deep, deep into the reservoir, a lot of it is basically permanently latent. So this concept that we can silence the virus so it doesn't come out ever is biologically plausible, happens in people. But as of now, there are no therapies. We've been playing around with mTOR inhibitors, these sort of cancer drugs. We think we're getting some modest effect, but there's not a lot of therapies in the clinic right now approaching this. This is the least developed trajectory in HIV cure, and I think one that's been ignored.
So we're looking for concrete still with that one.
We're in concrete on those cells, and we don't have it yet.
Okay.
But the nature does it, but we can't do it therapeutically.
Right. Cool. Linda, do you want to comment on it, or?
No, I think Steve's got it exactly right. We don't even have the water to put in the concrete yet, you know.
Right.
But I really do think that this is a really, I mean, it makes sense to me, which is like a joke sometimes because, you know, science often doesn't work that way just because it makes sense to me, right? But it just seems like in the long run a better approach to me. You know, of course, there's things like I'll get us started here with another area like the gene therapy that Steve described earlier, where people can maybe get one shot or one treatment and that's a wrap. And you know, at this point, maybe it's not scalable, maybe it costs $1 million, but just like antivirals, it used to be 3 pills once a day with food, without food, and all that craziness. You know, maybe we can develop the gene therapy into such a thing where we can go, you know, to Africa and give somebody a shot, and that'll be a wrap. I mean, I think that to me is probably one of the best and most promising all-around therapies, or in other words, approaches to deal with this. But then we got a lot of way to go to reach, you know, from to. But I really like the idea of you know, maybe something that you have to do a couple of times, but that is not every 6 months, even every 2 years.
So so Linda brings up gene therapy, which is a third trajectory, and this is one in which I think there's a lot of growth, and you know, it's a very broad area. You can genetically modify the immune system in many ways. You can surgically remove the virus, and your group there in Seattle is working on this. The problem there is that in order for that to work, you have to come up with a genetic therapy that gets every single virus in the body. But it's possible. It's been done in mice now. And so these are basically using vectors to deliver these molecular scissors that go around the body and just cut up the provirus. So that's one area. A second area is one that I think is more likely to work. Is do the same thing, but rather than cutting up the provirus, cut up CCR5. CCR5 is the host door by which HIV gets into cells. And we think this is possible because of Timothy Brown, the Berlin patient, and his brothers, I guess, in London now and Dusseldorf. So there are 3 people who didn't directly get gene therapy, but more or less got bone marrow transplants and were cured because of the concept that if you get rid of CCR5, you're cured. Then there's and so there's a lot of interest in getting stem cells and knocking out CCR5, and that's all very plausible, and we're working on that, and I actually think that actually will work. But that's going to be hard to make that scalable. A third area that excites us, in particular down in San Francisco, is rewiring T cells to make them very powerful to kill HIV. These are the CAR T studies that are happening now. And we think that this is going to work in large part because people in cancer are doing this all the time. And CAR T cells in cancer work. They're very toxic and they're very expensive now. But they're becoming much more scalable. Now you can make CAR T cells in a box that you can have in a doctor's office. I've seen them. And they're getting safer. They're not yet that safe, but they understand why they're toxic and they're making them safer. So that gene therapy in terms of knocking out the virus, knocking out CCR5, or making super killer T cells is an area of great interest to us and is a growth area in the field.
And it's our interest up here in Seattle, obviously, as well. It's one of the interesting things has been that toxicity, because right as we were plunging in, there was those Juno deaths that happened with brain swelling and CAR T cells. All of a sudden, like before that, I thought they were just like a godsend. It sounded like it was going to be a miracle, magical journey, and suddenly Larry Corey's having to deal with these deaths and finding out what it was. And I discovered the toxicity issues, which I hadn't heard about before then because they weren't really widely talked about at all. And now it's sort of interesting. What I like about CAR T cells is the idea, like Carl June this summer at our event that we did with him, was talking about people that he gave CAR T cells for HIV 25 years ago still had them in their bodies 25 years later.
Those are my patients. Those are my patients.
Yeah, see, and so
we did the first CAR T cell studies in the 1990s, and Carl's been studying them, and they persist for 20, 25 years.
Yeah.
And no one's grown a tail. Everybody's healthy.
Right.
They're much older, but they're healthy.
But that gives me heart that, like, if we do get that technology to work, that it could be a one-time treatment that would last for a really long time. And so you would have those ninja warrior cells now, you know, that you would be giving them that superpower to focus on the HIV infected cells. So even if it is latent for years and doesn't wake up, when it finally does, if something does it, you're going to have some warriors there to handle it. At least I know that's the hope. But so I'm biased, of course, but I also just, it appeals to me in that sense of taking care of a problem that we see may happen later, which is, how do you get all that virus? You probably won't. You know, 1 out of every million cells, how are you going to figure that out? You know, but to have something that's in your body for a long time that will last, I personally like that. I take heart that hopefully it will work.
Yeah, I agree with that. I mean, for the same reasons that you all described about gene therapy, something that we can maybe do once, twice, whatever, that's not going to drag people back, that people will be able to have access to eventually after a lot of work. But I love it. You know, HIV is an immune disease, so hello, that's what we should be looking at.
Mm-hmm. And the other thing I love that we're also doing up here in Seattle with Jen Adair's work, her gene therapy in a box, trying to make that scalability issue manageable, where basically if you have a plug that you can put in, you know, an electric socket right there, you can do gene therapy because of the way they've used this. They've repurposed this machine. So she calls it gene therapy in a box, but she just came back to our cab recently and told us that she didn't think gene therapy was going to be enough to make gene therapy scalable, because she was telling us a story about this 2-week motorcycle journey to a clinic for care. And then once the family got there for their child, they didn't have money to get back. And so she realized it was going to have to be something like a shot that you could take to them, because even to ask them to make a journey to a clinic sometimes was going to be enough of a barrier that would keep a cure from occurring. And so she's now a convert with the rest of the Gates Foundation and the other Defeat HIV researchers of trying to get that in a needle, which is the interesting part too, to think about that it's a needle that we're talking about, the end results. What do these look like at the end? What is block and lock going to look like at the end? Is it going to be a pill? Is it going to be a shot? Is it going to be some sort of microwave oven that you walk into? Who knows? But we know the last one is probably not likely. So we have 3 down, 2 more?
2 more. One is early art. So and the other is immunotherapy. So the big gorilla is immunotherapy. Let's just get rid of early art. So I always thought that giving early antiretroviral therapy in the first couple of weeks could be curative. And that's not true. You cannot cure HIV with treatment. But, and we've shown that, everyone's shown that. We have a guy in San Francisco that sort of started antiretroviral therapy on day 1 of the detectable infection, had no virus anywhere in the body, stopped therapy, 9 months later, it rebounded. Took a long time, but it was there. But, you know, we got this sort of group of people now out there, if they start therapy not super early, not in the first few weeks, but not late, not after like 3 to 6 months, but somewhere in between. You start therapy and you have the right genetic background, we don't know what that is, and you stop therapy and the virus won't rebound. These post-treatment controllers. All right, let's talk about the last two remaining trajectories. One is treating people in acute infection, and one is the big sort of gorilla in the room, or so to speak, which is immunotherapy. I'll talk about that last. But this concept that if we treat people during acute infection, we cure them. It first came by the Mississippi baby. You guys may remember several years ago when a baby was treated on day 1 and she was almost cured, but not cured. And then there's been a bunch of monkey studies showing that if you treat really early, the virus basically goes away. You don't develop latency. So there's been a lot of effort to get people on therapy in the first few weeks and see if we can cure them. Bottom line, it does not work. It may work in people. It will not work in. It may work in monkeys. It will not work in people. We actually had a case here in San Francisco of a gentleman who started therapy basically on the day the virus became first detectable. You could not start therapy earlier, and he had no virus in his body that we could find. And stopped drug 2 years later. Took about 8 months, but the virus did rebound, proving, from my perspective, you can't cure people. But I do think, and I'm a big fan of, the fact that if you start therapy early, not in the first week maybe, but like at around the first month, and go on therapy for a couple years and stop therapy, 1 in 10 of those people might not rebound, so-called post-treatment controllers. First identified by the French, but now confirmed by others. We don't know why this happens. We think NK cells are important. And more importantly, we don't know which of those 10 people, if you took 10 people and you stopped therapy, 9 would do badly and 1 would do well. It'd be nice to know who would do well. We don't know that yet. So that's an area of investigation. But this concept of post-treatment control proves that with treatment, you can modify how the body and the virus interact in a way that actually could lead to a long-term control or remission. I mean, Linda, what do you think about those observations?
Well, no, I think that's right on, and I think that you actually alluded to what the issue is there. What's different about that one person that was able to achieve post-treatment control as opposed to the other people that weren't? So for me, it feels like it's a host factor issue, and how do we get at that? How do we figure out what's different about that person? And I think that's the ball game in that particular strategy.
I think it's the exciting mystery of all of cure research, figuring these things out specifically. And since it already happens, quote unquote, naturally, it should be something that if we figure these things out, we might be able to induce in people if we figure out all the needed steps, and to even have markers of who might be a success without having to actually put all 10 people on a treatment interruption and have 9 of them not succeed just to get 1. Do you know where that
Michael, is a beautiful introduction to the full trajectory.
Cool.
Which is therapeutically trying to make people post-treatment controllers by modifying their immune system. Immunotherapy, and this is our thing. I know that, Michael, you guys up there in Seattle are gene therapists. We're more immunotherapists, although there's a lot of crosstalk now. But the whole focus of our group, and many others, mainly based on what's happening in cancer, chemotherapy, immunotherapy, and in monkey studies, is to bring people in who are on antiretroviral drugs, build up their immune system, focusing mainly on CD8 T cells, but looking at other parts of the immune system, and then seeing if we can achieve that post-treatment control phenomena. Let people stop, and the virus doesn't rebound.
And Steve, what do those CD8 cells do? I know people hear it all the time, but I don't think they have a good idea of what a CD8 cell does versus a CD4.
CD8s are the killers. CD4s are the generals, the captains. They tell everyone in the immune system what to do. CD8s go out and
do it.
Yeah.
Do it.
Cool.
So they're, but CD4s also, they sit on top of the whole immune system, and they tell CD8s what to do, but they also tell NK cells, natural killer cells, they tell dendritic cells, they tell B cells which make the antibodies,
Mm-hmm.
which bind to the virus. So it's very complex. But in the context of cure, we think the primary battle will be born between CD8 T cells and the virus in lymph nodes.
Mm.
So that's basically what we need to do. We need to find out a way to build up the CD8 T cells, train them to go after the vulnerable part of the virus, force them to go into the tissues where the virus is, and make sure that when the virus takes off, that they are in the right place at the right time, activated and primed to go.
Mm.
And to be honest, we're doing this with immunotherapy, but you guys are doing this with CAR T cells, same kind of idea.
Right.
But using gene therapy to make the CD8 T cells do what we're trying to do with vaccines.
Mm.
But anyway, so immunotherapy in general is kind of a hodgepodge of vaccines and vaccine adjuvants and antibodies and PD-1 inhibitors and CTLA-4 inhibitors and IL-15 and all this stuff.
Mm-hmm.
You can mix it all around. So then the monkeys, there's been multiple studies in which monkeys have been cured by some combination of all this stuff.
Mm.
And right now, the first combination studies in people have not worked. And not too surprising, the stuff that you can do in monkeys is far more intensive and aggressive than what you can do in people. We're starting off with some of the more benign, easy to do drugs. They're typically not the best ones. But we and others are now about to start some big, huge, well, not huge, but big, intensive studies involving products and drugs that we actually think might work.
Mm.
So that's my worldview on the 5 trajectories
It's awesome.
for HIV cure in 2020.
Uh-huh. And I just want to wrap up our conversation before it goes on too long, because we already did do almost a half hour in the first session. I just wanted to ask, what message about cure would you like to leave people with? What should they hold in their minds and their hearts from leaving this video and leaving AIDS 2020 and going back to their lives? What do you want them to think or to hold in their minds or hearts about HIV cure?
Don't give up. I mean, in other words, we're going to do this hopefully sooner rather than later, even if it takes a long time. This is a very, very, very awful time in the world. There's so many things going on, but we can't forget about HIV. We can't forget about the cure, and we just have to hang in there. I mean, you know, most of AIDS activism is holding the line, and you know, it's always going to be with us. There's always going to be work, and we just can't give up, and we have to keep going no matter what happens.
Thank you, Lynda. Steve?
I don't know. I think Lynda should have the last word. That was quite spot on.
You're letting her get off. We're going to get some wisdom from you one day.
My wisdom is the same. This is going to take some time. Be patient. 10 years plus before we have any real hints of something that's is a reasonable guess. So give us some, yeah. But Lynda said it better.
But I wanted to thank you both. You're both people that I love to see at CURE meetings. I appreciate the fact that you have a speaking style that is what I would call human and easy to relate to. And so I really appreciate you helping out both our cab as well as the DARE cab by recording this so we can have somewhat of a presence and hopefully help some of the world wrap their minds a little bit more around CURE research. So I wanted to thank each of you for participating in today's conversation. And I will be seeing you maybe very soon as we get closer to this, okay? So thank you, and take care, both of y'all. All right?
Bye-bye.
Bye-bye.
Bye. Thank you.